Gavin from Balls to You. So before we get into today's video, I want you guys to check out the description box below. All my information's on there, Morph Market, etc. And while we're on the subject of Morph Market, you guys can actually give me a follow on Morph Market. So what that means is the minute I upload something for sale, you guys will get a notification to see what it is I have uploaded, so you don't miss out. I'm getting lots of messages saying they missed out and there's nothing I can do because it's first come first serve. Obviously, hopefully you guys will follow me on Instagram. If not, get over there and follow me on Instagram. And also Patreon is proving to be an absolutely, for me, a real enjoyable place. The Patreon support is growing. I'm constantly adding new platforms to the Patreon, and there's lots of sensitive information that you won't find on YouTube that I do post on there to help you guys succeed with breeding and keeping ball pythons. So with that being said, I'd like to get into today's video. Me and this gentleman go way back, and I'm talking way back to the days of Ralph Davis forums, Reptile Radio, Herp Nation. For those who've been in the industry long enough, you guys will know those type of forums and those type of people from back in the day. Now, this gentleman, he kind of flies under the radar a little bit. His knowledge on genetics is nuts. He does know his stuff, and I'm not going to get too much into detail, but he really does. When he talks, I sit up and listen. So the gentleman in question is a guy called Travis Wyman. Now, you guys can give him a follow on Instagram. He's called Snakes and Bakes, or snakes_n_bakes. Give him a follow, show him some support. Trust me, what he doesn't know about genetics is not worth knowing. So with that being said, I'm going to shut up. Let's get into the video and sit back, enjoy, and I hope you learned something from this video, because I did. First of all, Travis, I'd like you to introduce yourself to people and tell people who you are and what you do and how long you've been in the hobby, etc.
Right. Well, I'm Travis Wyman. You may have encountered me on the forums in the past as Splendii. I'm on Instagram as Snakes and Bakes, and I've been in the hobby, well, like all of us, I've been in the hobby since I was a kid. I've been kind of seriously in the hobby for 10, 15 years or so. It's hard to really keep track after all this time. And by profession, I am a geneticist and a molecular biologist. And for me, that carries into the hobby just with how I view the animals that we work with, how I see, in the genes and the alleles and things interacting with one another. And so it's a bit more than just boom, paint job to me. It's, how these things work on their subunit levels.
Yeah. Okay. So for someone, I wouldn't say as stupid as me, but I'm not quite as intellectual on your level. So just explain to people roughly how we see pastel, for example. How does it express itself differently from the wild type, that sort of thing?
Yeah. Well, I will step it back and say it's not a matter of stupid. What I call it is ignorant. And most people hear ignorant and they take that as an insult, and it's not. Ignorance merely means a lack of education on something. You're ignorant of that genetic level, protein level, cellular level process. I'm ignorant of the finer parts of how a car's exhaust system works. I am ignorant of how the internal parts of a digital camera work. We all have some degree of ignorance in some things. It's not a shameful thing to be ignorant. You cannot know everything about everything. It's just not possible.
Apparently, people on Facebook do know everything about everything.
This is true. And, Facebook can be the downfall of humanity. But the only time I think that ignorance is a true problem is when you maintain a willful ignorance. When someone tries to explain to you, this is the way something actually is, you're making a mistake, and your answer is, no, you're wrong, because I say you're wrong. Not because I have proof to back it up, but because I'm just not going to listen to you. That's the only time I think ignorance is a problem, and that is stupid, to borrow back on the phrase. So nobody is stupid for not seeing things and viewing things the way I do. In fact, I predict that very few people see things the way I do just because of who I am and what I am. There aren't a lot of us in the hobby that have my level of background. Myself, guys like Nick Mutton. Yeah. Even though Nick doesn't have a formal degree, Nick has done so much research behind him. Warren Booth, he's a fellow doctorate. Ben Morrill, Justin Julander. These are all guys who've got a very deep understanding of it, and we look at the hobby a little bit differently. Yeah, it's not a better way necessarily. It just gives us a different perspective, and a lot of times when we try to share that perspective, we're trying to help educate everybody in the community. And that's one of the great things about you is I see you trying to educate other people in the hobby and in the community as well, and bring everybody in, try to get everybody on the same level or get them to better understand things so that they can all work towards these better goals with their projects, their animals. So going back to your question of what is pastel? Now, a lot of people will look at a pastel and they just say, this animal is more yellow. And fundamentally, yes, the animal is more yellow. I look at it as, okay, why is the animal more yellow? What is it about this animal that makes it more yellow? And so dig deeper. We know that there are two primary pigments to ball pythons. There's the dark brown-black pigment, which is melanin, and then there's that gold pigment, which is the xanthan. And that xanthan is actually a yellow pigment, and the reason it looks gold in our animals is because it's got the yellow pigment with the black pigment over the top, kind of shading it. And that black gold over the yellow makes it look, or that black brown over the yellow makes it look gold. Yeah. So what's happening in pastel is you're taking some of that away. Yeah. And now that yellow is showing through brighter. So what pastel is, in a sense, is a form of hypomelanism. You're taking away melanin out of a certain part of the animal. Now, this is different from what everybody calls a hypomelanistic animal as a morph, the ghost morph. Yep. Those animals are probably more properly considered hypopigmented because all of the pigment on them is sort of backed off. It's not just the dark browns, it's also the yellows that are backed down on them. So it's all of the pigment that's reduced on them, making them hypopigmented. But the hobbies called them hypomelanistic or just hypos for so long that hearing, well, pastel is a type of hypomelanism, a lot of people just kind of get this jerk reaction of, no, that can't be right. Yeah. And that's why, stop and think. If I pull that black melanin pigment away, hypo is just less than, so it's less than the normal amount of black pigment. Yep. And that's also what gives you that extra blushing and stuff is in some of the places, the black pigment is a lot deeper and heavier. And in some places it's only in lightly. And when you pull away that, one layer, if it's just stacked layers, if you pull away that one layer, then you lift it off a bit. It's kind of like pulling a gauze layer off. Now you can see some of those blush spots a little bit better.
Yeah, and I suppose would that also work with little traits? For example, if you make a super pastel, the head's more blushed.
Yeah.
So would you say, or would I be right in saying that obviously we know a super pastel is two pastels, if you like, lying on top of each other?
Hmm.
Is that a similar sort of thing? So in other words, what you're trying to say is if we take more away, which is what a super pastel is, we're taking more of that layer away, which is making it more blushed, more yellow, more faded. Is that,
Yes.
yeah,
So it's you can think of, I like to talk a lot in analogies because I think it makes it easier to visualize sort of mentally.
yeah,
The the pastel gene is basically, like I said, it's like a lace doily.
yeah,
Okay. And you have, because it's a chromosomal thing, it's two genes, one from mom, one from dad. So you have two lace doilies of the dark pigment that the pastel gene controls. So if you take one of them away, now you take one layer of that lace doily away, and what's underneath shines through some. And then if you take both of the lace doilies away, what's underneath shines through even more.
yeah,
And the
yeah,
pigments aren't just painted on like a single layer. They're painted on in, multiple layers. So you've got, a black layer, a yellow layer, a black layer, a yellow layer, a black layer, yellow layers. And that pastel, those lace doilies are in there somewhere, and you're pulling them away, but you still have all those other layers in between and underneath that show through.
okay. So for those who are watching this, this is mind blowing, and this is also very interesting because for me, again, it's like what we said. It's not just putting a snake together and making a paint job or a pattern paint job or a combo. For me, when I listened to you back in the day, it was more of trying to understand how a particular gene works. So, how does that work? Why does it work that way? Why does it react like that with, let's say, leopard with a lesser or with a bell complex? Why does it do that? Why don't other things work that way? So that got me thinking. And I believe you spoke about a chimera.
Mm-hmm.
Did you speak about? I believe you did. You did a little bit about a chimera or how a chimera works. Was that?
Yeah. So
Yeah.
a chimera, we often see in the hobby what we call paradoxes.
Yeah.
Okay. And paradox is it's a bastard term to the hobby.
Yeah.
Paradox has no legitimate scientific value in the genomics circles. If you go calling paradox to anybody, any non-herp geneticist, they're going to look at you like you're crazy because they're not going to understand what you're talking about.
Yeah.
Now, what we call paradoxes are just animals that show sort of a paradoxical pigmentation. An animal that should not be showing splotches of albino and normal, or both splotches of spider and normal, or pinstripe and normal, or lesser and normal. And there are a bunch of ways you can get paradoxing, but the two main ones are chimerism and mosaicism. And so how that works, a chimera is where you basically get two embryos and they fuse together, or parts of them fuse together. And if those two embryos are completely different genetically, morph-wise, then you see expression of both genotypes in the animal. So if you mix an albino embryo with a wild type embryo or a het albino animal, then the full animal looks mostly or partially albino, but then has the black and gold spotting of the wild types. Or, the inverse. It's mostly black and gold like a wild type, but it also shows the full patterning of an albino. And that's because it's got two different genetic payloads. Now, mosaicism works in a somewhat similar way, except it's only just from one animal. It's just that you have some type of localized loss of genes, and it'll give you that same thing. So if I have a het albino animal, but in a small spot of it, it loses that wild type gene that balances out the albino, then in that just little spot, it'll show the full albino phenotype. Or, if you've got a yellow belly and it loses the wild type, it shows an ivory phenotype in that spot.
Yeah. So, which is interesting, because when I heard you first discuss and explain the chimera theory regarding how we can get like a paradox, it was quite interesting, because every time since then, I think that was 2010, possibly a little bit before that. I can't remember, but when
I've discussed it so many times I couldn't tell you.
When I've heard you talk that, every time I see now an animal with a heavy paradox in, that's something, exactly using your analogy, like a normal, but it's showing albinoism, I sort of think, and I look at that and I think, is that a normal het albino, or is that an albino, but it's showing the wild type from underneath? So it kind of, again, listening to you from back in the day, sort of makes me now look at a combo, or look at an animal when something appears out of Africa and it looks really weird. I look at that and I go, no, hang on, that's probably just a paradox animal, or that's just probably just a chimera effect, if you like. So that's, for me, when I take little things from what I've learned from you, and I believe that we should all sort of listen a bit more and take more in from people like yourself, because we could learn a hell of a lot, and just trying to understand how something works. My background is, I've worked in the motor industry since I was 15. I'm 40 now. And if you said to me, Gav, how does a DPF work? How does an EGR valve work? And I can explain that to you, and I can make you understand. If you understand it, you can then look back and go, well, actually, my DPF can't be playing up on my car because it's only a filter. So in actual fact, it's a result of a cause. So the cause is the EGR valve playing up. The result is the DPF is blocked.
Mm-hmm.
So that's when I listen to you and you say, well, the result is a paradox animal, but the cause is this. And that's what I find interesting. And one of the things I find interesting at the moment, that I'm seeing a lot of people hatch parthenogenesis. Have I said that correctly?
Parthenogenic.
Yeah. That one.
Parthenogenesis is the term for the action, but when a clutch happens, it is a parthenogenetic clutch. Yes.
Parthenogenetic clutch. Yes. There you go. Even I'm struggling to say. So I'm seeing quite a few people hatch those type of clutches. Now, I've hatched, I want to say, 3 over the years. And within those clutches, or what I'm hearing is, it's a partho clutch because they're all females. Now, I'm going to tell you what I think, or what I logically, with my diagnostic head on, and sort of the way I think things. If there was one female snake left in the world and Mother Nature said, no, you're not ready to be extinct yet, you're going to lay a clutch of eggs. There's no male around, but you're going to lay a clutch of eggs. To me, it makes no sense. This is what I don't understand. It makes no sense to lay a clutch of females without a male, because we all know in reptiles and mammals, mating in the wild, they don't know who's mom, who's dad, who's brother, who's sister. So for me, I look at it and I think, well, if a snake's going to lay eggs in the wild, surely there's got to be a male in there, because out of the 3 Partho clutches I've had, 2 of those clutches have had males in them. And I'll give you the one from this year. So this year, I bred a fire butter to a which was the female, and I bred it to an orange dream vanilla. Now, the orange dream vanilla was a proven breeder male for Aussie, so he's proven to have vanilla, and it's not fire. And then the mom was definitely a fire, because she came from a super fire. There wasn't no retained sperm. This was the first clutch she'd had. She gave me 3 viable eggs. One of them was a butter, and the other 2 were super fires. They were black-eyed leucistics, pure white snakes. One was a male, one was a female. Now, they, in my eyes, a super fire, you tend to get a couple of yellow splotches up and down the body occasionally, maybe. These were pure white, and even though their eyes were black, they weren't solid black. They were kind of like a dare I say, like a bluey black,
An indigo,
almost as if it was a super fire, super butter, right? And it threw me, because I'm hearing, and this isn't the first time. The first clutch that I had with Partho was females. The second clutch, again, similar sort of thing, proven genetics on the mother and father. The mother was a virgin, and again, I had a Partho baby. Which was a super form. It was a super pastel, and there was no way that the dad had pastel on the other side, because again, he was proven to be just a leopard. So he wasn't a pastel leopard, he was just a leopard, sorry. So this clutch, now I've hatched a male and a female, and of course in my head I'm thinking, well, that's standard. If it was in the wild, it'd need a male to continue to mate with the other females that have just hatched, and to pop, for the species to continue. However, I'm hearing, I hear a lot of breeders say, oh, if they're all females, then it's got to be a Partho clutch. And it's kind of being used really fluently, if you know what I mean. They're sort of using it, they're throwing the term around. And I just wanted to speak to you and sort of say, well, I know a guy who can answer that question for me, whether something's not right my end, which I've done what I think you would do, which is go over the breeding, look at the parents, confirm the parents' genetics to make sure we've not got a gene floating around that I've missed. And I'm still coming back with this male and female thing. So do you want to help me with this and shed some light?
I'll do my best. Okay, so a parthenogenetic clutch would be all females, and that's comes down with the nature of how these come up. Visual aids here. Everybody's, got their chromosome count, and you have 2 copies of every chromosome. One from mom, one from dad. Okay? And it's easy to see that when you're just thinking, wild type, fire, pastel, wild type. Each of those covers. And when you breed, you only pass one of them on. So you either pass on the fire or the wild type allele there. And then depending on what you get from dad gives you how it's going to pair up. Okay. Now that's the simple end of how we look at it. But we need to look at it at a little bit more depth, which is how do we get our sperm cells and our egg cells? And so that starts with, your basic genome, where you've got 2 copies of everything. And then those cells first undergo a doubling effect. So everything in them gets doubled. And then the cells divide and get half of the genetic payload for each gene, each chromosome. Okay.
Yeah, that makes sense.
And then these divide further to give you a sperm cell, a sperm cell, a sperm cell,
Yeah.
and a sperm cell. Okay. Now, that gives you 4 sperm cells, each carrying 1 copy of each chromosome. And then those go on to match up with an egg, which would also have 1 copy of each chromosome.
Yeah.
Now, the way that the egg cells divide is slightly different. With the sperm, you start with 1 cell and you end up with 4 sperm. When you do the eggs, you start with 1 cell. It does this first division.
Yep.
And then the cell that has these falls away and dies and leaves you with just this.
Yeah.
And then it divides again, and the cell that has these shrinks down, and basically if this is the cell, the egg that you're left with, it's the size of a grapefruit. And this cell with this genetic payload shrinks down to the size of a pea and sits on top of that grapefruit. And is basically just a trash thing. It's called a polar body.
Okay.
And this is mostly inert most of the time. Now, what happens in parthenogenesis is something causes this pea to react and get absorbed back into this egg. And that gives you a full complement.
Okay.
But if you started with a mom who is a fire, and she's carrying the fire allele and the wild type allele, that first division that got rid of that wild type, but leaves you with 2 copies of the fire, and then that splits. When this joins back, now you're left with 2 copies of the fire gene, and that's what gives you a super fire. Okay?
Okay.
Now, the same thing happens with the sex chromosomes. If mom is carrying the male and female sex chromosomes, and when they first divide, when they first divide, the male sex chromosomes get tossed. All you're left with is the 2 female ones.
Okay,
so
that makes sense.
If you had an egg that was left with the 2 male ones, and this was the polar body and it tried to fuse back, this genetic combination is non-viable, and the embryo dies. So when you have a parthenogenic clutch, all you can have is the females left behind, the XX females, because the YY males, those embryos die, they never develop. And that's part of the reason that parthenogenetic clutches tend to be smaller than normal clutches. part of it's also because you have essentially massive inbreeding happening within the mother, and some of those inbreeding events can create deleterious mutations that cause the embryos to fail.
Yeah, within the mom.
Within the mom, yes.
Yeah, okay.
But when all you're left with is that female genotype that's viable, then that's all you can get is all females. Now, sometimes you can get cases where, instead of seeing, it's easy to see a parthenogenetic clutch where the mom is a fire and she makes super fires.
Yeah, of course.
And go, okay, so this must be parthenogenetic and they're all female and I got super fires and normals and whatever was in mom. But sometimes you get a clutch where most of the animals hatch as you would expect, but then say dad is a pastel and mom has no pastel in her, and you hatch a super pastel. And it's female. And everybody's like, well, how does that happen? That's a different form of not parthenogenesis, it's called androgenesis. And it is essentially the male parthenogenesis effect, or parthenogenetic effect. What happens is that the sperm, instead of that second division happening that splits it apart into two individual sperm cells, it doesn't divide right. So you just get one sperm cell carrying that double load.
Yeah.
And when it manages to hit an egg cell, if that egg cell is a null and doesn't have any genes in it, it will carry out like this. Or even weirder events, just a single sperm hits an egg cell, but that egg cell is a null. It activates it, and then in the egg, it doubles the whole thing over. So you get a half clone of the father, and it carries forward that super pastel phenotype because the father had a pastel, but you got the doubling of only his genes because the mom's genes weren't present at all. Now, how you got a weirdo male is definitely something different.
Yeah.
That would argue against parthenogenesis.
Okay.
And there are a lot of different ways that could occur.
Yeah.
But the most likely thing would be something really simple where, say, in the first division of that embryo when it was developing, the wild type allele just got lost. And so then every cell after that only had one copy of the gene, and it was the fire copy, the fire allele. And so the animal looks like a super fire, but genetically it's still sexually produced. It's just missing that one gene. And it's missing that one gene because, there are different ways it can happen. You can lose a whole chromosome.
Yeah.
Your chromosomes are basically shaped like a string of beads, and maybe half of the beads broke off, so you're missing half of those genes, or something weird happened there.
So would you say then that particular male, because you mentioned earlier on that the males, if that happened and you were to get a male, it wouldn't be viable. It wouldn't make it out the egg. It wouldn't die.
In a parthenogenetic event, the male wouldn't make it out alive.
Okay.
Yours, obviously, you got a male, so it wasn't a parthenogenetic event that created that male. It was something different.
Right. Okay. What was really bizarre was the two females, the one super fire and the one butter, did really, really well. Really well. The male didn't do too well. So in other words, he started, well, he wasn't eating straight out the gate, but the two females thrived. Now, in my eyes, in the wild, maybe that male wouldn't have made it, but obviously I had to force feed him. And he's switched on now. He's smashing food. He's doing fantastic. I've sold his sister. She's due to go out next week. I've got the male on Morph Market, but I'm really tempted to keep him and just do a little experiment and breed from him and see exactly kind of like just to take it that step further. I mean, I obviously you've explained it to me and I understand it. I just want to. I just always thought that if a species was in the wild and it was close to extinction, that the female, that the female would lay a clutch of eggs, and when they hatched, there'd be a male in there to help with the
to help recover the population.
the lost. Yeah, because
And
there was
that's sort of an old way of thinking of the reason behind parthenogenesis was the last ditch species survival effort, and that way of thinking was because people thought it was a really rare type of occurrence that these parthenogenetic events would happen. But the more people have looked into this phenomena, people like Warren Booth, he studies this extensively. If you go to his website, Booth Lab, he's got a bunch of papers listed there that detail this. Now, they're all scientific papers, so they're really heavy on the science jargon. But if you go into that and read in depth, one of the things that you'll see is that parthenogenesis is not as infrequent as it once was thought to be. He was able to detect it in wild snakes at what would be considered a ridiculously high level if it was this weird freak thing. It also occurs not just in like a full parthenogenetic event. You can have clutches that are split where some of the babies are sexual produced animals and some of them are parthenogenetic animals. And if that happened, you'd never really know because you'd get a clutch with both males and females. And, assuming you're just looking at a wild type of clutch, you wouldn't know that some of those females were parthenogenetic animals. So that sort of takes away the impetus of saying that the only time parthenogenesis happens is when females have no males around, but they're trying to preserve the species by making more of it in the hopes that if a male does come around, there'll be a whole bunch of females for him to be able to then breed and reestablish the species.
Yeah.
And now just seems to be a sort of little understood reason why, but not uncommon thing that it happens. The ultimate reason why it happens is still not fully known, but it doesn't seem to truly be this genetic last ditch effort type of thing.
Yeah, because obviously we know retained sperm can happen on some instances, etc., etc. And the reason why this kind of subject leads me into something else that I read about a month ago. So there was a ball python. I don't know if you read this. It was at St. Louis Zoo in Missouri. Did you read that? Did you see that? Yeah, it laid eggs.
Yeah, and they said she's like 67 years old and she's been in captivity for something like
she's
62 years.
Yeah, she was 62 when she's laid this eggs. She's apparently not been in contact with a male for over 15 years. And while some reptiles are known to reproduce asexually, which I believe Komodo dragons do that, don't they?
Yeah, it's what parthenogenesis is that's just asexual. So Yeah, the Komodo dragons do it. We've seen in ball pythons. It's been seen in retics, Burmese pythons, carpet pythons, copperheads, cottonmouths, garter snakes. I mean, we see it all across.
And quite a few species, So why would this female, do you think, what's the purpose for this female laying eggs, doing the parthenogenetic clutch? Any ideas?
It's just this thing that happens,
Okay, so it's not.
The conditions were, there were some conditions that were right that triggered her to go into a full breeding cycle.
Yeah.
What those conditions are, we haven't fully determined. We can explain the mechanism behind what happens, but we don't know the why of it.
Okay, so this leads me into another question, which there's kind of two sides. Some people sit on one side of the fence, some people sit on the other side. So when we talk about breeding ball pythons, and we talk about when a female is growing her follicles, now some breeders say they wait till their female is, I don't know, 15 millimeter plus before they put a male in.
Mm-hmm.
And some breeders will say, actually, if you don't put a male in, it won't spur the female on to grow follicles. Is this something, because obviously I know you breed as well, and do you see anything in this? Because I'm on the side of the fence where I believe a female ball python has to have that physical interaction, i.e. the male going in with the female, and I believe that helps the female grow the follicles. What's your take on that one?
I think it's going to be different for every individual animal. I think in some cases it may help.
Yeah.
That said, I had a couple of years ago a female that I wasn't planning on breeding and had not seen a male all season long. And when I was cleaning tubs, I went to put her back in and I was just letting her crawl over my hand. And it wasn't an intentional palping, but I was able to palp her, and I felt, basically 6 golf ball sized follicles in her.
Yeah.
And like I said, she hadn't seen a male. She hadn't been near a male. The male, I keep my males in a different rack than my females.
Yeah.
None of the females around her had been paired, but she had those follicles. And I was just like, well, what the hell? And I grabbed a male and I threw him in with her. And 2 weeks later, she ovulated and went.
Yeah.
So obviously it didn't matter to her that she hadn't been exposed to a male until that one lock that she had and bred and went. I have another female I have bred. I have paired her. Can't say I've bred her. I have paired her every year for 6 years. And she has yet to go. Doesn't matter how many times she sees a male, I can feel the start of follicles. I get those little blueberry, cherry-sized, and they never get bigger than that. So even though she's seeing a male every single year, she doesn't go. I don't know what's up with her. I don't know why she just doesn't want to go. But obviously seeing a male makes no difference to her.
Okay.
So I think it's down to the individual animals.
Yeah.
And, obviously in the wild, that could make or break which animal is going to breed. In captivity, it's not as big a deal. If the female didn't need to see a male, but you're putting her in with a male every year, and she goes, it doesn't matter. If the female needs to see a male, and you're introducing her to a male, and that's what triggers her to go, she goes. So now both of those females go, whether or not they needed to see a male.
Yeah.
Personally, I would think males that don't require seeing a male in the wild would probably be more genetically fit, as it were, because if they can be ready to go the instant they stumble upon a male, in that chance that it happens, then they can breed right then and there and go on and produce their offspring.
Almost
Versus
be
if there's
ready
a
to go.
female who first has to be introduced to a male a bunch of times before she's triggered to go, well, she might see a male and pair with him, and that might start her going. But if it was a bad pairing, or she, while sperm storage can happen, maybe she didn't store it right or whatever, and then she doesn't see another male, she got triggered to go, but then she didn't have the follow-up good lock and pair to actually reproduce, then her genes don't get carried on.
Yeah.
So it's
it's weird, because the other thing we have to consider as well is most of us, we have a reptile room, so those animals are all in the same room, and if people who feed frozen thawed, if they defrost their frozen rodents in the room, by the time they're defrosted, you walk in, you can see the snakes.
every snake is already
Exactly. Yeah. So with that being said, I suppose we could be creating that kind of smell, even though the female's not seeing a male. She's smelling something. It's in the air,
yes,
It's something there.
she's being exposed to the pheromones and the hormones,
Yeah.
not just of the males, but of the other females that are being paired in the room, and those ones that are coming. And that could be part of it too.
Yeah. So that's something, again, for me, when I look at, like, I had a female exactly the same as you said. She wasn't eating. She was a good weight, and her cycle. So I got her from a friend of mine, and her cycle was, she would be laying eggs around about within this month, for example. Now I thought, right, okay, she's gone off food. Her breeding cycle, because I'm a big believer, if you do buy breeder females, that you buy them from a reliable source, and you kind of find out their cycle, their lay cycle. So at least you can try and work with their cycle and fit in around their cycle, if you like. And she was due, and she'd gone off food, and she was a good weight. And I thought, you know what, I'm just going to put a male in. I didn't ultrasound her. I've got an ultrasound. I didn't ultrasound her. I thought, I'm just going to try it. Put the male in. Sure enough, she started smashing food. Literally, like you said, within 2 weeks, she ovulated. This female ovulated in about 3 weeks. And sure enough, she's due to lay eggs in the next 25 days, something like that. So I also looked at that, and I learned, and this is something that I always try and get my subscribers or viewers to sort of pay attention to their animals, learn their animals, potentially grow your animals up from a hatchling and find out their characteristics. Because if you buy, unless, and I don't mean this in a nasty way, but unless you're an experienced breeder, you've got a few years under your belt, it's very difficult for a new breeder to come in and think, I want to breed ball pythons. I'm just going to get a male and a female. I'm going to breed them. I'm going to produce eggs. What's difficult to explain to that person is you don't know what that male and female are like. You've only literally brought them. You want to breed them. So how do you know when her cycle is? How do you know? You've just brought her off a breeder, and all of a sudden she's gone off food. Oh, she's gone off food. Okay, but it could be through a cycle. She could be going through a cycle. She may not want to feed, because I find ball pythons aren't exactly greedy snakes. They're not like a boa or a retic where you could feed them every day, let's say. I find there's a point where they stop feeding because they're full, they've had enough, whatever. So if you don't understand the cycle of that snake, it could create some false illusion that, oh, that snake's not eating this, that, and the other. So for me, understanding the cycle, especially if it's a female, understanding the breeding cycle for me is important, because it's what's going to make you a successful breeder, if that makes sense. As in, not a lucrative breeder. Like when I talk about success, I don't mean loads of money and loads of clutches. I just mean
Just the ability to be
a
able to do it.
successful. Exactly. It's just a successful breeding process and understanding why a female's acting that way. Why is a male acting that way? Why is this? So, for example, you can go in sometimes and pick up a male and be cleaning, put him back. You'll go in and pick up another male, put him back, and then you'll go in, you'll pick up another male, and the other male now can smell these other two males' scent on your hand, and all of a sudden he starts kicking back on you. You know, he just
Mhm.
starts, "Huh, huh," like get off. And again, it's understanding that. Well, yeah, I understand it. I've got the scent on my hand. Just because we don't see it doesn't mean it's not there. And
Right.
I think there's a lot of things that we miss as humans that we're not paying attention to our snakes, which would help us if we did pay attention. It would help us a hell of a lot more. And part of you talking to us was I wanted to get across to people and say, look, there's more layers to these species than just a pastel color, an albino, a pied, a clown. There's a whole nother level that's out there that you can learn if you wish. And sometimes it makes for it gives you such a greater understanding that when an animal does something or you produce something, you can now sit back and go, now I understand. I remember Travis, and like I said to you at the beginning, I remember things you talked about, and I was like, okay, that's interesting, yeah. And so that's in the back of my mind. So when anything occurs, I can look at it with an open mind and go, okay, why has that happened? Why is this? Why is that? And I think I believe personally having you on is as important as speaking to Justin or speaking to Ozzy about morphs and projects and this, that, and the other, because this is a side of the hobby which a lot of people don't delve into. The genetics. Why does this happen? Why is this? Why is this happening? And a lot of the times it can be guesswork, but a lot of the times it's because we're not paying attention to what's going on with our animals, and that's my take on it. And that's why I feel I have more of a success rate, because I do pay attention. I try and understand things, like I said in the beginning, from a diagnostic. Why is my car misfiring?
Mhm.
It's not, oh, it's got a misfire. It's lacking power. Why? What's causing it? Like I said, there's a cause and a result. And it's the same thing. If you feed your animals well, you learn, the result will be, fingers crossed, if you've done it correctly, a healthy, viable clutch. And this is something that, like I said, getting you on, Travis, just to have a quick chat and talk about this, I hope people will take from this and sort of people will stop and think and go, well, this is really sort of interesting, and there's more layers to this, and there's more to learn. You know, um,
It's to borrow from Eugene Bissett.
Eugene, he's
Basically
in, yeah,
summing up everything you said, you become a student of the serpent.
yeah,
If you're not paying attention to your animals, you're not going to be successful. And it's not just in breeding, it's in everything. I mean, if you're not paying attention to your animals, how do you know if maybe your temps are off? How do you know if maybe your humidities are wrong? How do the animals will tell you if something changes, if something's different. If a storm front rolls in, you can tell by the way your animals behave. Like with the food, yeah, your animals all change. They all come up to the front of the tank. Well, what happens if you go in and only half of your animals are up at the front of the tank when you're thawing out food? Why is that?
mm-hmm.
Maybe there's something going on with those animals. Is it one whole rack that's off and not coming up to the front? Is that because that thermostat on that rack blew out and all of those animals are cold?
Yeah.
And so they don't want to eat because they know if they eat while they're cold, it's going to be detrimental to them.
Yep.
It's It's that student of the serpent. You need to pay attention to your animals in all ways, big and small. And that's how, like you said, that's how you become successful, not just in breeding, but in keeping.
Yeah. Yeah. I mean, it's exactly the same. I mean, I've kept different species over the years, and you, again, just sitting there studying and watching them, at night, in the day, why are they hiding during the day? Like, certain species of dart frog, and why are they hiding during the day? Why aren't they more, I want to see them. Where are they hiding? And there's certain species of the dart frog which are more brazen and will come to the front and they're like, hey, look at me. And then there's some that are like, oh, I best hide. There's a big scary monster outside. And it's sort of learning the species. And this is where sometimes people make the wrong mistake and they think, look at this cool looking snake. And then they keep it a few years and they actually realize that it's a reticulated python or a Burmese. And all of a sudden they can't keep it because it's too big.
Mhm.
So I always say research, research, research. Research before you get the animal, research the animal when you have it, obviously pay attention to it, and then try and learn a bit more about where it's come from, its habitat. Like, as well as I do, ball pythons do extremely well in rack systems.
Yes.
Um, and I don't get it now, but I think we've educated enough people now where you used to get, oh, why are you keeping them in rack systems? That's cruel. That's this. Why are you keeping them in little tubs in the dark and this, that, and the other? And I think because we know the species and we're still learning. Like now, the freedom breeder tubs, the freedom breeder have done, which are the FB40s, are even smaller than the FB70s. Why is that? Why is that? Because they like smaller, tight spaces. They prefer that way. I have one female, which she's 2015. She's a proven breeder, and every year I go to breed her, I put her into a breeding tub. The minute she locks with a male, she will start smashing food in that big tub. She'll lay a clutch of eggs. She'll have one meal in that big tub. The second meal, she won't bother. The third meal, she's not interested. I have to take her
Down
out,
factor.
yeah, into an FB10 tub. So you've got this 1200, 1300, because she's only a small female. She's 1200, 1300 grams crammed into this FB10. But the minute you put her in there, that night she will start smashing food like a boa. That's one female out of 70 odd females, let's say. So when every one of them's different. And if you're not paying attention and you're not learning and you're not willing to, like you said, become a um, what was it you said?
Student of the serpent.
Student of the serpent. That was it.
And that's, you got to credit Eugene Bissett with that. But
Oh
it's,
yeah, yeah,
it's one of those
Eugene.
things that sticks with you when you
Yeah,
really click with that idea. You just,
yeah,
it's there.
yeah, yeah. Well, I've kept you long enough. We've been talking for about an hour now. I really appreciate you coming on, Travis. It's been
Not a
absolutely
problem.
awesome. I think what we ought to do is I'd like to do. I missed you used to do the 60 seconds genetic.
The 60-second genetics on Herp Nation Radio, and then
Yeah,
unfortunately went the way of the dodo as well.
yeah, yeah. So for those people who don't know, Travis has obviously been around. I'm not going to tell you his age, but he's younger than what he looks. And he used to do these 60-second genetic uh, a little section on Herp Nation Radio, and it was fantastic because those 60 seconds was. I remember always having to turn it up a little bit just to listen and sort of rewind it back and listen to it again, and then oh yeah, yeah. It's really interesting. That's why I've always had a huge amount of respect for Travis. That's why I've always looked up to him. Because he's someone that I used to listen to And I used to, when Travis talked, my ears plucked up because for me, I wanted to learn that next level. I wanted to understand how things work. And I'm still learning. I think, I hope I never stop learning. And as long as we've got people like you in the hobby, Travis, um, it's going to be good. So we ought to do a bit more of this. I think maybe pick a topic, maybe not too long, maybe keep it short. Maybe we get some questions thrown in from the viewers that we can look through and go, oh yeah, that's really interesting. Let's discuss that maybe in future. Maybe we can hopefully educate people in a positive way.
Well,
So I just want to thank you for coming on and taking time out of your evening. I really do appreciate it, Travis.
it's not a problem. I appreciate you
Okay.
having me on, and I appreciate what you had to say about me. I'm glad that I have been able to help you. Um, it's it's kind of humbling and flattering to hear that coming from you. I mean, I like I said, I see what you do as a huge benefit to the hobby.
Thank you.
You know, you're very big on getting information out there to people, and it's a very positive information, and that's a huge thing in my eyes.
Yeah.
So being brought on here to be able to share with you means a lot to me, and I appreciate that. And yeah, I'm happy to get together with you in the future. You know, if there's other things that people would like to know or that you'd like to know, we can
Yeah.
figure something out. And
Definitely. So for anyone who aren't or isn't following Travis on Instagram, it is Snakes and Bakes.
yes,
I'll
snakes
put a link,
underscore n underscore bakes.
yeah, I'll put a link in the description box below. Hopefully you guys will show him some support. Go and give him a follow. The guy, trust me, his snakes are amazing and his food looks even better. Maybe we can do some cooking classes. I don't know. But yeah, absolutely awesome. So get over there and give him a follow. Make sure you show him some support. And if you do drop him a message and you did hear it from me, make sure you say Gavin sent you and you appreciate his help with this little subject. So, but for now, hope everyone is all okay. Take care, and I'll see you guys in the next one.
That money clip us now, the new now looks like. Whoa, upgrade her diamonds, now her girlfriends are like. Whoa, 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 whoa. I hit record a job, you can't ignore it. I'm transforming.